I think it's happening.
Yes, yeah.
Wow. I think it's happening. Excellent. Okay. Apologies, everyone. We had some significant technical issues today. I'm not going to go do a long intro because we're already about 15 minutes past the hour. Um, Again, this is the Fine Seminar Series. I'm Lauren Hayes. I'm today's host. And today I'm really happy to have Asher Leeks speak to us. Asher comes from the University of British Columbia as an assistant professor there. He did his PhD in evolutionary biology at the University of Oxford with Stuart West, held a visiting position in Japan, and then had a postdoc in Yale at Yale University in the States before taking on his new role in British Columbia. And really quick, I was just excited to have Asher come speak to us because he's going to be talking about viruses, something that, I only think about usually when I'm giving a lecture to my intro bio class. And so it's going to be really interesting to hear how viruses can sort of fit into the theme of the social evolution seminar here in the Fine seminar. Um, Again, I'm excited to have Asher, and I would give a longer intro, but today, Asher, let's just get you going. And I'm thankful you're here, and thanks for everyone else for being with us today.
Absolutely. Thank you so much, Lauren, for the invitation to speak here. It's a fantastic seminar series, and so it's really wonderful to be here. I'm just going to go and share my screen if that's okay. Very good. And okay, so if this is coming through, then I'm gonna start.
It looks great.
Perfect. Okay. So yeah, so I often call this talk the social lives of viruses, and I think as Lauren alluded to, speaking in a seminar series like this, you're presumably more familiar with more traditional kinds of social organisms, things like mammals or birds, or increasingly microbes, but perhaps not at the all the way down to viruses. Typically, when you think of viruses, if you open a textbook, you think of a kind of solitary viral particle that kind of acts on its own, causing a disease. What I'm going to try and convince you of today through a couple of different stories is that viruses can not only be studied within the same kinds of tools and frameworks that have been developed for 100 years or so in social evolution, but also that there are some ways in which viruses are actually more social than more traditional study organisms. Okay, so first of all, what do I mean by viral sociality? And then we'll go into two different studies at different timescales. So when we say viruses being social, at least for the purpose of this talk, I'm primarily talking about social interactions that take place within an infected cell. And the key to this is that the two fundamental things all viruses need to do is replicate their genome and then package that genome inside a virion or a capsid, which is that typical kind of spiky ball image that you see. Both of these processes require gene products that can be shared, at least in principle, among multiple viral genomes inside the same host cell. So replication enzymes, like this little Pac-Man here, typically that can replicate any viral sequence as long as it's got the right recognition bit. Similar with capsid proteins. So this means in an evolutionary sense, we can think of them as public goods. You pay some cost to encode them, which we'll come onto in a moment, but the benefit, importantly, can be shared among multiple genomes, not just the one that encoded it. So we previously reviewed kind of the scope of these interactions. And so I've mentioned these within cell shared gene products, but you can also get some really cool interactions among viruses that are infecting different cells and at the host level. So there's a whole kind of like world out there of essentially virus behavioral ecology to explore. But for the purpose of this talk, I'm actually not going to focus on all the kind of cooperative things that they do. Instead, I'm going to focus on the dark side of viral social interactions, which is the possibility of exploitation by cheats. And when I say cheat in this talk, I'm primarily talking about viruses that have come from the cooperator genome and they've been deleted. So kind of spontaneous deletions, which are quite common in many viruses, quite error-prone replication, that deletes the bit of the gene that codes for the shared protein, the public good, but keeps all of the other bits, keeps the recognition sequence, keeps the bit that gets you into the capsid. What this means is that these smaller genomes can benefit from the public goods without contributing to them. So they can arise easily, kind of de novo. And importantly, because these cheats are not encoding the gene, it's not just that they avoid the cost of producing something, but actually their shorter genome gives them an intrinsic replication advantage a lot of the time. So you can imagine that over multiple rounds of replication within a cell, this advantage can start to compound. So what you often find in viruses is that these cheats get really extraordinary advantages, sometimes orders of magnitude fitness advantages over the course of an infected cell or cell culture, which is much bigger than what we tend to see from cheats in other organisms. So we previously kind of drawn analogies between different types of viral cheats and other cheats that you see kind of in nature. And I like to show this slide because I think what this shows is that although this language of using social evolution and virology, as you pointed out, is new, actually the traits we're describing, a lot of them you've known about for a very long time. So the very first attempts to grow viruses in the lab were this Danish guy, Preben von Magnus. And what he would find with influenza viruses, he'd find these cycles. And it turns out that these cycles are caused by the spontaneous generation of these cheat mutants, defective interfering genomes, that then spread, suppressing the viral population, which then leads to a rebound, and you have these kind of ongoing oscillations. They're reminiscent of Lotka-Volterra kind of interactions. So this is something that crops up every time you try to grow a lot of animal or plant viruses in the lab. You run into this problem. Or if you don't bottleneck the population, these cheats invade and can reduce the stock. And there's interesting parallels as well. So things like DNA origami and phage display often rely on synthetic cheats that they've created by essentially deleting all of the public goods genes for M13 filamentous phage, and that gives them a backbone that they can then fill in that genome for other purposes. So really, I think what this shows is that viral cheating is not just something that we're kind of that we're inventing. This is something that the virologists have been rediscovering for nearly 100 years in different areas of virology. Okay, so one of the first things that often gets mentioned when you start talking about viral cheats is people will ask you, okay, well, that's great. Does this mean it could be used as a therapy if it's so interfering? And maybe 5 or 6 years ago, I might not have been so confident answering that. And just to be clear, the idea here is that if you're infected with a virus, we could manufacture a synthetic viral cheat. So that's a reduced version of the virus that doesn't have the public goods genes like the replicase, so it can't infect a cell on its own. The idea is if you infect it into a viral infection, the cheat might replicate at the expense of the cooperator, eventually take over, and then cure the infection. Very similar in theme to something like phage therapy, except here we're not really using a different species, we're using a kind of inherent social interaction. Now, there's actually been a burst of interest in this really since COVID, and the kind of take home from this figure is basically that this type of therapy is surprisingly effective for a broad range of different viruses. A couple of different modes of action. Sometimes it's direct kind of cheat-based interference. Sometimes the cheats have an immune response. All of these studies are in animal models. So this really is kind of progressing fairly quickly. This timeline I put in for my fantastic postdoc, Rowan Green, so I think it illustrates the kind of timeline on this. The first of these cheat-based therapies, this Pichai et al. 2024 paper, this is actually now in stage 1 human clinical trials. Okay, now my sense on cheat therapy is that a lot of the questions that you would want to know before you really start rolling it out or doing it more broadly are the evolutionary questions. How are viral cheat cooperator interactions going to evolve over the course of an infection and then over longer timescales? And so now, and I argue that these kinds of clinically relevant questions are also exactly the same questions that we would want to ask if we are behavioral ecologists or social evolution theorists thinking about viruses. So now I'm going to go through 2 stories of how these viral social interactions can kind of help us understand things about how viruses evolve and about viral infections. Okay, so the first story is going to be about something called a multipartite virus. Now, I think a lot of us, when we're drawn to biology, particularly in a seminar series like this, we're drawn by the crazy adaptations that life can have, and in particular these social ones. Things like murmurations of starlings, right? Things like birds of paradise. Things where you look at it and you just think how could that evolve? Now, I'm going to argue that multipartite viruses fill this role for a lot of virologists and for a lot of people like me who got into virology a little bit later. So to explain what a multipartite virus is, most viruses are what we call monopartite, which just means that their genome encodes the things that they need it to do, which seems like a sensible strategy. Multipartite viruses instead split apart their genome, and each of the physically different genome segments then transmits in a different capsid, and all those capsids need to infect the same cell, or at least the same host, for the virus to then be reconstituted. Now, I probably don't need to hammer the point home too much, but this is a really costly strategy. Because even if you have a high rate of co-infection, there's a chance that all of the viruses infecting the same cell will have the same segment, not the right combination. So just on the face of it, this seems like it's not obvious why this strategy would evolve. Despite that, the first multipartite viruses were discovered in the 60s. It's evolved among multiple different types of genome organizations. So we think it's evolved independently many, many times. Accounts for something like 20% of plant viruses. So this is not rare, this is a common strategy. And in some cases, it's not just splitting into 2 or 3 different particles, but Faber-Green and necrotic stunt virus has 8. So the combinatorics of trying to get 8 different particles into the same target at the same time is really challenging. That's a massive, massive cost. So the challenge as evolutionary biologists is for us to explain, why does this exist? So there are some existing ideas about multipartitism. A lot of them focus on the idea of group benefits or species level benefits, which is where our minds often go to for this kind of thing. So there's a little literature which tries to figure out, if you're a multipartite virus, might that give you some kind of collective benefit? Say if maybe it means your genome segments can reach different frequencies in different hosts, which might help you kind of genome regulation. Or perhaps it means that you can have smaller virions, which might give you an advantage, like they can survive longer, for example, sometimes. The problem with these kinds of explanations is that they often require quite specific mechanisms about the viral capsid or the kind of hosts. So it's not obvious if they apply super broadly. And then the really big problem with them is that the benefits don't scale the way that the costs do. If you think about the costs of splitting up, every time you split your genome from 2 to 3 to 4, the cost doesn't just go up incrementally, the cost starts going up super linearly because now you have a harder combinatorics problem. So for these benefits to explain multipartite, and particularly the highly multipartite viruses, they really run into a problem because the scaling doesn't work out. And so it's hard to think of a mechanism at the kind of group benefit level that would scale in that superlinear way. So there's good evidence for some of these arguments, but my thinking on this is that these are super important for the maintenance and why multipartite sticks around, but they struggle to explain the origins. Okay, so I'm going to suggest that cheating could provide a mechanism by which multipartite viruses can evolve. And the inspiration is pretty simple, that when you look at a multipartite virus and each of the segments only encodes one gene, that looks a little bit like these viral cheats we saw earlier, which are deleting genes but keeping others or keeping the other bits. So the question is, can you go from one to the other? And the idea that I'm going to put forward was first put forward by Sean Nee in 1987. And so to be clear, what's happening here is we're going to imagine that you start with a population of cooperator viruses that encode 2 genes that can both, in principle, be complemented. We're going to see what happens when one type of cheat invades, and then a second. And we're going to say that multipartite evolves if you can if the cheats can replace the cooperator, if the final form of the virus only has the genome split up and doesn't have this like monopartite form. Okay, so does this work? So the first thing we can do, just go through the model setup. We're going to assume viruses don't co-infect at some rate, so just a single infection, in which case the cooperator can replicate, the cheat can't. If they co-infect, it's a little bit more complicated. Two cooperators can produce a productive infection. If the cooperator and the cheat co-infect, the cheat gets a benefit, it replicates faster. Two cheats of the same type will fail, but two cheats of different types can in principle complement each other and get some replication governed by this parameter E. So this gives us a payoff matrix in the double infection case. So for the values that we end up using, this basically looks like the hawk-dove game, except that we have this third player and the possibility of a defector-defector interaction. So this gives us our replicator equations, which are pretty straightforward to solve. And I'm going to show you here the kind of classic dynamics of what happens when we play this out. So in the low co-infection regime, if everyone infects a cell on their own, then the cooperatives just win. We expect this. As you start to crank up the high co-infection, you start to enter a world in which the cheats can invade. These gray lines here are the cheat populations, assuming they're symmetrical with these two cheats, starting from low frequency and beginning to invade. And you'll notice here that it goes to an equilibrium. We expect this from a hawk-dove game. The difference in this model is when we crank up this complementation possibility, this E parameter. When we crank this up, then this frequency dependence can get broken. Because you enter a world where if these two cheats are high abundance and the cooperator is rare, actually the cheats can meet each other. So they don't need to meet the cooperator, which means you break the frequency dependence and you can actually have a world where the cooperator fully goes extinct and you just have the cheats. So this is the multipartite world. Now, how hard is this to get in the model? One of the nice things about these kind of super simple models is that you can draw out pretty clear conditions. So this right-hand world exists, provided the likelihood of complementation, this E parameter, is above a threshold that's defined in terms of the co-infection rate, the ratio of viral single to double infection productivity, and this C term, which is the cost that the cooperator pays for interacting with the cheat. So in other words, if the cooperator and the cheat are in a cell, how much does the cheat crush the replication of the cooperator? Now, what we then did is we went into the literature and we found experiments that people had done on defective interfering genomes of known viruses and tried to infer these A and C parameters. And then that lets us determine how hard would multipartite be to evolve in that system if you had two complementing cheats that looked like a known cheat in the lab. And what we see is this. So in the blue region of this diagram, you're getting the multipartite virus invading and the cooperator going extinct. In the white region, the monopartite form, the ancestral cooperator, is surviving. And what you can see is that this is happening at pretty low co-infection rates. So for the properties of poliovirus, you only need about half the cells to be co-infected. So that's an MOI of about 1.7 or so if you're a virologist. So this is not a particularly hard condition. And it turns out this is true for a wide range of different viruses, at least when you look at defective interfering genomes. The important thing is that these defective interfering genome cheats tend to have a strongly negative effect on the replication of the cooperator, which means your C value is low, which means it's fairly easy for multipartite to evolve. Okay, I also mentioned this collective benefit idea that people have put forward. Well, we can look at that in we can look at one way of doing that in this model. Below this red line, we're assuming E is less than 1, so we're assuming that cheats complementing each other in a cell are getting less productivity than the cooperator would. And in all those plots I showed you, there always exists like a little blue part of the plot underneath the red line, indicating we're getting a transition to multipartite, even though it's intrinsically costly. So we can actually do this more formally. We can calculate the population fitness before and after. And it turns out that there always exists a value of E for which the multipartite population can invade and the cooperator goes extinct, that doesn't require that the resulting population is more fit. So in fact, it's always possible in this model for cheating to derive multipartite viruses without that requiring a group benefit. Now, the group benefit it could be useful because this could help us to explain these highly multipartite viruses that I mentioned the collective benefits arguments kind of struggle with. If you're not requiring a collective benefit, maybe cheating helps you go further into those highly multipartite transitions. What I'm going to show you here is an agent-based simulation where and this becomes stochastic at this point. So what you're going to see is each line is going to be several hundred simulation runs, and I'm going to color in each line with the fraction of outcomes that lead to either monopartite or multipartite to different degrees. And this is a model with 8 genome segments and no intrinsic benefits. And what I want you to look at is this kind of middle right half of the multipartite plot, where we're getting transitions to multipartite viruses with 3 or 4 segments at reasonable multiplicities of infection. So MOI is just the average number of viral particles infecting each host cell with a Poisson distribution. So less than 10 is kind of within the realms of kind of realistic possibility. And again, what we're seeing is these 3 or 4 multipartite viruses can invade, whereas without cheating, the typical group benefits models tend to require MOIs really astonishing values, up 100, 1,000 or so for those numbers. So the important thing is that because we no longer require the population to be doing better to predict multipartites, we can kind of blow open the parameter space and predict it over a much wider range. Okay, so the final little part of this story is whether there's any empirical support for this prediction. And it's a little bit difficult to test because multipartite viruses typically evolved a while ago, and kind of proper comparative work can be difficult in viruses. But what I'm going to show you here is 5 different kind of viral realms, which we're pretty confident are independent evolutionary origins of viruses. And I'm just going to show you the kind of fraction of genera for which we know that they get cheats of the right type on the X, and then the number of genera that we know to be multipartite on the Y. And we can draw this kind of like straight line through a few points. Not enormously compelling, but is at least consistent with the idea that cheating is important for multipartite virus evolution. Just to walk you through that, we showed that cheating can drive multipartite in a way that doesn't lead to a collective benefit, which in turn helps us explain multipartite virus evolution for the higher N. Okay, so that gives an example of where we can take reasonably standard thinking within social evolution and really formalize a kind of new way of explaining an age-old idea within virology. What I'm going to switch to now is I want to talk about social conflict emerging not over long evolutionary timescales, but instead over the timescale of a single infected host. So I mentioned that these viral cheats are kind of everywhere, and this is true in the sense that there are lots of different types of viral cheat that people have seen, especially when growing viruses in the lab. But there's a bit of an open question as to how common these cheats, in particular these defective interfering cheats, these ones that kind of pop up spontaneously, it's been a kind of long-running open question as to how common they are in natural viral infections. Again, we see them in tissue culture. Do we also see them in hosts? So what I'm going to show you here is our attempt to start to ask about whether during the kind of infected time course of an acute viral infection, in this case, we're going to focus on influenza, do you see these cheats emerge, or do we see signs of them emerging, and what are they doing? Okay, so because cheats of this type, defective interfering genomes, because they're formed through large deletions in the public goods genes of influenza, this makes them quite predictable, which means you can look in the sequencing data and we can find individual viral reads where they keep the end bits, but they span a large deleted region in the middle. And so my fantastic collaborator Andrew Routh has an algorithm that's kind of designed to do this. And what you do is you essentially kind of go through the viral population. And here, all these different colors in the tablet viewer are different viral reads. And when you see these kind of long stretches of white, that's telling you a single read is spanning a large deleted region. And when you throw up sequencing data from, say, influenza passaging, for example, and you look at it, what you see is that, in particular for the polymerase genes, you see these white bits in the plot kind of like they go all the way down the screen. You see this massive proliferation of viral reads that have got a massive internal deletion, specifically in the polymerase segments. You tend not to see it for the other segments. And there's been quite a lot of work now in the lab, kind of really kind of perfecting these methods for detecting them and really establishing that predictably the PA, PB1, and PB2 genomes of influenza are susceptible to these cheats emerging. Now, the data set that we're going to look at here is from the H5N1 avian influenza outbreak, which is currently all over North America. It's most famous for infecting cattle and chickens, but it also infects a super broad host range of birds and mammals over a couple of year periods. This gives us a super wide diversity of both flu genotypes and host species to look in. And so in this data set, we've got more than 10,000 sequences, each of which broadly comes from a single infected animal. And this is all publicly available data that the USDA have made available. So the first challenge is taking this algorithm that Andrew's made and adapting it kind of to work at this scale, which a graduate student Praneeth did, who's in Christian Anderson's lab at the Scripps. And so we kind of applied this to all of these sequences in NCBI. And there's two assumptions that are kind of important before I show you the data. One is we're assuming that when we take a swab from these animals and sequence it, we're assuming that that swab reflects kind of the whole viral population within that animal at that time point. And we're also assuming, because the bottlenecks are quite narrow in influenza, we're assuming that each infection started with a single influenza cooperative genotype. So if we see any cheats, we're inferring that they evolved de novo and they didn't come with the cooperator at the beginning. And all of the next work that I'm going to show you was done by Rowan Green, who's a fantastic postdoc who joined my lab about a year ago, just after I started. Okay, so the first gene I'm going to show you is the non-structural protein for influenza. I should have mentioned, just to be clear, influenza has 8 genome segments. You can kind of think of them like chromosomes, but they're all inside the same virion. So it's not multipartite, but it is segmented. And this means we can do this analysis for each 8 of these genes separately. And what we see here in this heat map is we're filtering just for deletions that delete more than 30 nucleotides of the coding region. So we're pretty sure these are non-functional. We're pretty sure they're deleting the typical coding function of the gene. And this is just pooled across all 10,000 samples, and this is count data. So we're seeing that for the non-structural protein, you have this kind of reasonably uniform splodge, which we can contrast with PB2, which is one of these 3 segments that we know get cheats. And what you see is this really dense spot in the top left of the plot. So that's showing us that we've got a very high count of deletions that start at the beginning of the genome, delete all the way to the end, and then rejoin at the end. So this is so these guys have deleted 80% or so of the coding region of the polymerase, and they're keeping really just the end bits. These are molecules only a few hundred base pairs long. And you can see this crazy high density. This is a log plot, so it's slightly underselling it. And we see the same pattern broadly across PA, PB1, and PB2, which are these 3 subunits that make up the polymerase. You don't see it quite so clearly in the other segments. You can get a little bit in HA and NA, which are capsid genes, but typically it's these polymerase ones that are really dominating the density. And one kind of take home from this is across all 10,000 animals, we're finding almost 1 million unique deletion containing viral genomes. So this is an enormous pool of genetic diversity that's being generated in these infections. And if you kind of sum up all of the deleted reads, this bar plot here is kind of stacking. This is count data, but this is not just count data. This is weighted by our estimate of the abundance within the sample of each of these deletions. And what you can see is that more than 90% of the big deletions we're finding in this data set are deleting more than 75% of one of the three polymerase subunits. So of the deletions that influenza is kind of generating, the ones that are reaching high abundance are overwhelmingly these large deletions of the polymerase subunits. Again, this is strongly consistent with what we see in the lab when we see these defective interfering genomes emerge that delete large regions, get an advantage because they delete it, and then spread. Okay, so that's kind of at the aggregate level. I'm now going to zoom in and show you some data at the level of each individual host. So on the X here, we're going to see all of the samples in the data set kind of put into their percentiles. And on the Y, I'm going to go through each of these 8 genome segments, and I'm going to estimate the kind of total abundance of large deletions for each segment. And there's two things that kind of stuck out when you do this. The first is this left hand of the plot. So this gray line is just which individual has kind of the highest defective load among all of the 8 segments. So if we take 1% relative abundance within host as our cutoff, which is a normal cutoff that might be used for a minority variant, basically every individual in the sample has these defective fairing genomes at 1% or higher. So they're almost ubiquitous. But the crazy thing is that in about a third of individuals across this whole data set, these defective viral genomes are not just there, but they're exceeding 50% of the relative abundance. This means that they're outnumbering the cooperative wild type. And remember that these are deletions that are deleting more than 30 nucleotides of the coding region. These are not typically functional genomes. It's pretty safe to assume they can't initiate an infection on their own. In social evolution language, they're obligate cheats. So we're finding that obligate cheats are dominating the population, as best we can tell, in a third of individuals for influenza. So we can also break this down by genotype of influenza and by host taxonomy. So here, so D1.3 and B313 are two different lineages of H5N1 that have kind of radiated after this zoonotic spillover event. So this is the same host group. This is landfowl, so things like chickens and turkeys. And there's about a 5-fold increase in the proportion that are reaching this 50% cheat dominance line if you're infected by D1.3 instead of B313. Same when you look at host taxonomy. So this is D1.1, which is the clade for which we have the most number of different species. Comparing waterfowl and landfowl, it's pretty similar. So it's like ducks versus chickens, basically. But when you look at hawks, the figure changes here. In hawks, more than 2/3 of them appear to be DVG dominated. So there's a very strong host effect here. We don't know if this host effect is to do with something to do with host biology, or if it's to do with something like the time of sampling. It might be that the hawks are typically sampled later in the infection if they're hunting bycatch, whereas the chickens and ducks might be sampled earlier if it's a routine surveillance. But there's a very strong signal in the data. And this comes out, Rowan has a kind of beta regression, and it turns out that viral genotype matters, host taxonomy matters, and there's also an interaction. R squared overall is approaching 10%. And so here you can kind of see the interaction. So you can see that actually in a few cases, the kind of B313 D1.1 effect flips. In terms of overall trends, it's not, we can't do every possible comparison we would like to because there are limits in the number of samples available from each group. But when you're comparing 313 to D1.1, B313 seems to have a lower cheat abundance than D1.1 when we can make the comparison, which is consistent with what we were seeing earlier with the DVG50 line. And you also see these taxonomic trends. So things that eat meat, so cats, canids, and these skunks and stink badgers and things, these seem to have quite substantially higher DVG loads. And then we find the opposite trend for things like ducks and cows and that kind of thing. again, I don't want to overinterpret this data too much because there are multiple compounds that might be going here, but it is a very strong signal that pops out of the data. And so this is true for cheat abundance, comparing these hosts and genotypes. It's also true when you look at cheat identity. So what I'm going to show you here, so Andrew had the idea of comparing this problem to kind of single cell RNA-seq data. So you can kind of imagine that the influenza kind of infection is kind of like a genome, and these different cheat mutants that are being generated are kind of like different transcripts. So we've borrowed from UMAP, and what we can see is here we've just kind of thrown all of the hosts in, and you can see there's a very strong genotype trend that you can see, which is actually orthogonal to the direction which cheat abundance is covered in. So this means it's not just cheat abundance that allows separation between hosts, but actually that different host species are getting different specific sequences of DVGs reaching higher or lower abundances. This signal is most pronounced when we focus on these polymerase segments. Again, these are dominating the deleted genome signal. This works both at the genotype and host level, but importantly, if we break down just to D1.1, we can get a separation into different host species purely based on the defective genomes and their abundances in the sample. The reason this matters is if these DVGs are reaching high abundance in infected hosts, it's plausible they could be being shed into wastewater, for example. This gives you some hope that you might be able to, if you were sampling and sequencing wastewater, you might then be able to infer the infected animals that were shedding into it, which will give you a new kind of public health tool for tracking. You can go further than this, and you can start to pull out individual DVG sequences that are over abundant or under abundant in different groups as well, which will be the next thing we'll do. Okay, now something you might be thinking, especially this time of year, is that it's not just animals that get the flu, right? It's us. So I have a collaboration with Kirsten St. George at the Wadsworth Center in New York State, who's been very kind and gotten us a couple of hundred sequences from seasonal influenza outbreaks from between 2014 and 2017. And Rowan did the same analysis on that, and it turns out that for humans, our best guess is that we're 2/3 likely to be cheat dominated. So if you got the flu and you got sampled in this data set, there's a 2 in 3 chance that the average influenza genotype inside you is not the cooperator virus that we think of, but actually it's this fully defective genome that we infer can only really spread through exploiting the wild type genome. For humans, we seem to be right on that right-hand point of the natural variation, 2/3 cheat dominated. So the next thing we're going to do is kind of break this down, and we're going to see if this cheat abundance predicts clinical outcome. If it does, that would be helpful for these therapeutic studies I was mentioning, and also might give you a biomarker for predicting the outcome. Okay, so just to sum up this second story, and then I'll conclude. So we looked at about 10,000 natural individuals from a broad range of different species, and we found that sequences that look like they come from cheats were basically ubiquitous, dominated by deletions in this replicase enzyme, influenza public good. Overall, in a third of individuals, cheats dominated in a way that depended both on the flu and the host, and for humans, the figure is 2/3. So we would argue that this indicates that cheating is not just a lab artifact, but actually it's a pretty huge component of natural influenza infections. Okay, now this is a little bit crazy, right? If in a third of individuals these cheats are dominating, that seems like it should be selected very strongly against at the level of the influenza kind of genome. So what's going on? There are some ideas that there might be some benefit to the influenza genome from having cheats around. Maybe it slows down the infection, which means you get more transmission opportunities, that kind of thing. I feel that those arguments often require quite a lot of assumptions about the kind of the epidemiology of the infection, about relationships between virulence, viral load, that kind of thing. And I'm gonna make the argument that I think our null model here should be that these viral cheats are basically like cancer for viruses. And this is actually not just my idea. So David Baltimore and Alice Wang have a review from 1970 where they define this idea of defective interfering genome for the first time. And when they define this, they suggest that the generation and spread of these cheats could be a naturally self-limiting part of viral life cycles, the same way that the immune system is inside a host. So one possibility here is that you're getting infected by flu is replicating, the cheats are spreading. If flu transmits before the cheats reach high abundance, which it might do because they take a few days to reach abundance, we assume, then actually flu might be able to just kind of jump from host to host, generate a bunch of cheats. So what we're sampling here is kind of the end of the lifespan, essentially. So this is a kind of more dynamic approach of thinking. And one way of thinking about this is that you've then got two things going on. You've got selection acting on the wild type influenza genome, which influences the generation of these cheats. And then once they've created, you have this kind of co-evolutionary process where selection is acting on the cheat genome and on the cooperator genome over that duration of the infection. Okay, so hopefully I've managed to convince you that it can be useful to use some of these ideas from social evolution to study viruses. And one way I like to think about this is that when we open a textbook and we see this kind of image of a virus, instead of seeing it just as a kind of solitary particle doing its thing, I think instead, at least in influenza, it looks like what a viral infection is a diverse community where social interactions are taking place, that really kind of dominates the makeup of the population. So I'd like to thank various collaborators for this on multipartite, on flu, and on social viruses, particularly Rowan and Leah, who have kind of joined me at UBC, and various funding sources. If you're interested in these ideas, we have a conference which this year we're hosting in Vancouver, Canada, in August. For this conference, we fully cover all accommodation and food and registration costs, so it's only travel that you have to pay for. And if you're interested, kind of get in touch and I'll send you a link to register pretty soon. We like to have these meetings, a big mixture of people doing virology and different viruses, as well as evolutionary biologists who can bring perspectives on how to study social evolution in other systems. Thank you.
Thank you, Asher. That was a really fascinating talk. Can you hear me okay now?
Yes, all good.
Great. Thank you again. And so this is an opportunity now to have some discussion. I'm really excited to hear what people are thinking. For those of you who are new to the Find, please, if you have a question or comment, please put a question mark in the chat and I will call on you and direct you to Asher. If you're on YouTube, please post your question directly in the chat function there and I will ask it for you. And if there are students and postdocs out there, we really want to encourage you to, ask questions. So I may move you up in the queue. So it's an opportunity now to have some discussion. So please go ahead and start dropping the question marks in the chat. Michael, please go ahead.
Yeah, thank you very much. I don't have a background in viral biology, I'm afraid, but what I found particularly interesting was these multipartite viruses and to understand how that comes about. And my question is whether there is any evidence for a kind of dynamics of division of labor between these different particles. Would that be an explanation why this comes about? Because they're more efficient in encoding for different functions when they split up.
Yes, and one way that the division of labor might come about mechanistically is one of the ideas I very briefly mentioned, which is when you have the same multipartite virus in different plant hosts, the ratio of the different segments changes, and it seems that the kind of number of copies that you need of each differs depending on the host. So it kind of gives you a kind of like division of labor. I think there are two issues with it. One is some of the benefits of division of labor you could get from being segmented, not just multipartite. Like if a shorter genome segment is better, you could just split them up and have them in the same virion like flu. And then
Yeah.
the other really big issue I have with it is it's not obvious to me why the benefits of dividing your labor are going to be, 10 times better to do 3 than 2, or 100 times better to do 4 than 3, or 1,000 times better to do 5 than 4, which is what you really need when you get these kind of accelerating costs of every additional split.
Yeah, it would be an extreme factor, beneficial factor. Yeah. Okay, thank you.
Yeah. I think one thing that is possible, like when we study multipartite viruses, we're studying the derived state, right? We're studying them after thousands of years of evolution. So it's, which for a long time is for a long time in viruses. So one possibility is that they do now have these kind of mechanisms of division of labor and regulation, etc. It might be that by splitting up, you set the stage for kind of new things to evolve that weren't possible with the single genome. But it's still, I still think that all of these mechanisms, if that's what you're in, if that's the idea, are it requires multiple steps. It doesn't give you like an initial kind of drive to divide.
Yeah. Okay. Okay. Thanks very much.
Thank you.
Next we have Perfect, please.
Hello, everyone. Thanks for the nice talk. I just have two questions before I go to the field. So the first one, I wanted to know, like, whether if viral cheating can actually like drive the evolution of new virus types or new infection strategies. That's the first one. Then the second one, do virus social interactions follow the same evolutionary rules as cooperation in animals?
Thank you. I love that question. So the first one, so yes, but not necessarily. I'm not aware of examples of viral cheats evolving in a way that then leads to a new kind of like proper viral lineage that is self-sufficient. So it definitely drives the origin of new strategies for spreading. So something we're starting to learn, for example, is some viral cheats. So there are some species of virus that essentially depend entirely on being cheats. So most humans are infected with adeno-associated viruses. It turns out these adeno-associated viruses, they kind of hang out in you. And when you get infected by an adenovirus or a herpes virus or something like that, they can kind of hitchhike on it and use it to help themselves spread. So these types of viruses, they often have quite elaborate strategies for exploiting like a range of other different host types. So it's not obvious if this is like really like a novel function, but it is at least novel for that lineage. This, and sometimes they'll be quite elaborate. So sometimes they'll do things where they'll take the capsid proteins made by the cooperator virus, and then they'll encode something that will restructure the capsid to primarily encode their genome instead of the cooperator. And then you've kind of got like a new capsid being created from the previous subunits. So bacteriophages, some of the cheat species do that. And then again, yeah, you could argue that multipartite viruses potentially might evolve new, they might evolve a higher host range if they have this genome formula thing that might be going on. But again, that's a couple of steps removed from the cheating. The second question, do they follow the same rules? That was part of the initial kind of excitement for me in starting to get into viruses. I think there's the way that I think about it is I think that viruses exist in maybe an extreme area of parameter space relative to the types of organisms that we usually study. So the benefits of cheating and the costs of cooperating can be really extreme in viruses. In 3 passages, you can get viral cheats. That's just like a few generations. You can get viral cheats going from creation to dominating the population. That's a really rapid selection pressure. So in the rest of social evolution, I think we typically see cooperation as kind of the trait, and we often model cheating as like the lack of that trait, right? We say, okay, you're a cheat if you don't do the cooperative behavior, the cooperative action. Viruses are, I think, are a little bit different because as soon as you stop doing the cooperative action, firstly, you can get these massive advantages. So it means that you're instead of being kind of like some minor fraction of the population, I think you can have this much more dynamic situation of kind of repeated invasions and extinctions and kind of less stable equilibria. And then the other thing is these flu genomes or other things that make these big deletions. The creation of it is through a deletion, so that's a typical evolutionary process. But then you've got this other genome that's got entirely different kind of selection maximans, entirely different fitness landscape. It's kind of like a different species. So I think that we should expect this conflict in viruses to be far more common. I think we should expect to emerge a lot and spread a lot and see it a lot more. And then I think that the co-evolution between the cheat and the cooperator acting on both becomes really important for the outcome in a way that for normal social evolution is sometimes less important.
All right, thank you very much.
But
And thanks for the nice job also.
Thank you. No, and I'm fascinated if you have any kind of insights or comparisons to things that you or other people study, be great to chat about them.
Okay. Thanks.
Sophia, please go ahead.
Hi everybody, my name is Sophia. I come from Adriana's group. So I have two questions. One is mainly about ecology, and so I don't know if you can answer that one because I know the topic it's like from like another line. So I would like to ask if maybe both viral genotype and host taxonomy influence the abundance of the defective genomes, and how might the ecological host factors such as like immune pressure or transmission bottlenecks interact with viral social dynamics to shape a cheating evolution?
It's a fantastic question, and this is something we would like to look into next. So there are some obvious predictions you might make. So if you're a host that has a super strong innate immune response, and so your infections typically get cleared in 4 days, you would expect a lower cheat abundance than all else equal than a host which has long chronic infections that can go on for weeks. Because in the second one, you've got more of an opportunity for cheats to be generated and spread. So one obvious prediction that we might make is that the duration of infection, which is a result of immune response, I think body size also predicts this, that should matter. Another thing that should matter, which I think you're alluding to, is the ecology of the transmission. So if you are if you're a host that has, say, like super dense kind of flocking behaviors, if you're like a broiler chicken or something, you might there have a situation where the same individual can be infected by multiple other infected individuals at the same time, which might increase the likelihood that cheats could spread between hosts, which might then mean that we see more cheats because you're not just starting from the origin, you're starting with some abundance. Those are two obvious predictions. The extent to which we can test that in this data, I'm not sure, because a lot of this was kind of sampling data. It wasn't necessarily from a designed experiment. And then I guess the third thing that comes to mind is we know from lab studies that if you passage the same viral genotype in different host organisms, so either closely related species or even in male and female of the same, or male and female guinea pigs, for example. The specific cheats that get formed and the abundances differ between those two. So because it's like a mutational process, it's likely that various host factors that are too complicated, I think, for me to speculate on, are going to affect the specific kind of likelihood to different types of mutation. So you might have a kind of like canalized path depending on, on the host resources that the virus is using.
Okay, thank you. And the other question would be if cheating genomes can drive genome fragmentation and the evolution of I don't know how to pronounce that, multipartite.
Yeah.
I'm not sure. Viruses, could multipartism itself be interpreted as an evolutionary strategy that stabilizes viral population against the spread of full cheats?
That's a really, that's a fantastic idea. And I'm just gonna pop to a slide that shows exactly that. So, if I can find it. But basically the answer is yes. So, in the simulation, we actually extend the simulation and we have not just the cooperated the cheats that delete genes but keep some, but we have the cheats that delete all of the genes, so they don't encode for anything, which is that full cheat that you're talking about. There was some back and forth between Lin Chao and Sean Ni about this in the 90s about how much this destroys the model. When we looked at this in the simulation, it basically turns out that yes, fragmented genomes have a lower abundance of full cheats than the cooperator. So yes, the second implication is the nature of the split actually matters. So here you can see, comparing the curve in this one versus these ones, you can see that the full cheat abundance is way higher in the monopartite case than the multipartite case. But then there's variation within the multipartite. It turns out this variation comes out from the inequality of the split. So if you've got 8 genes and you split to 4 and 4, you're more vulnerable to full cheats than if you've got 8 genes and you split to 7 and 1. The reason is that if it's 7 and 1, then on average, your successfully infected cells are going to have 1, 7, and then a bunch of 1s because the 1s are replicating faster, which means you've got a much more competitive environment for the full cheat to exploit. So yes, the effect is real. The caveat to the effect is I think it's a small advantage relative to the costs of multipartite strategy in the first place. So something that's kind of like hidden in the simulation is the fact that the actual population level abundances in the multipartite versus the monopartite case are like orders of magnitude lower. It's just like a way worse strategy. So it does protect you from full cheats, but I don't think that protection is enough to overcome the costs of splitting in the first place. It's like cutting your nose off to spite your face.
Okay, so a teeny more like a small question, like a follow-up question, is that does that mean that they have a certain type of trade-off between, let's say, the segments they have and the amount of replication they can make, and how much damage does that cause to like the hosts itself?
So you asked about the effect on the virulence for the host.
Yes.
Yes. Yeah. So presumably being multipartite, yeah, it's kind of funny because the way that I'm constructing these models is I'm assuming that the maximand is just like spreading, right? I'm assuming all else equal, the virus wants to successfully infect cells. Obviously, the implication if it's spreading faster is that's more costly for the host. And really these viruses have like two components to the fitness, right? There's the spread within host, and then there's the between host effect. And it's possible that spreading slower within the hosts can be better for between host spread, a classic virulence transmission trade-off. And there is some thinking that viral cheats either might be less costly or might be helpful, and that same logic would extend to multipartite if they make you less virulent within the host. I think there's two issues with that. One issue is it's not obvious why doing cheats to reduce your growth rate is better than just growing slower. You could just we know that replicase can vary and how fast it is. You could just reduce your replication rate if that was what you were trying to do. And then the second thing is that viral cheats are quite a chaotic way of doing this, because as soon as you introduce another agent that's evolving in the system, that's got some evolutionary pressure to wipe you out, essentially, to exploit you as much as it can. So it seems like it's very difficult for selection to tune it as a virulence control mechanism. And in particular, multipartite is really costly from that respect, because multipartite makes it really hard to infect new hosts. So if you're these virulence trade-offs are often assuming that a virus inside one host can just hop to infect a new one. If your genome is fragmented, there's this additional assumption where not only do I have to hop to infect a new one, but the vector carrying me, or whatever the transmission mechanism is, has to have multiple particles, and they have to be the right particles. So I think if you did the numbers on it, I think it would be hard to make it work.
Yeah, that's all. Thank you very much.
Thank you.
Next we have Karsten.
Yes, I'm Carsten Schradin, researcher at the CNRS in Strasbourg at the moment in South Africa. You already nearly answered my question just now, but I'm still asking it. It's a great talk, extremely interesting for me, also a bit frustrating to show me how stupid I am. But what you just said, I mean, basically, if you have infection from host to host, it cannot be just by cheats, because alone they cannot survive. But from what I know, I think from the discussion about the corona pandemic, is that normally infections from host to host never go by one single virus, but by something that is called a virus load. Can you tell me how much does such a virus load approximately have to be? Is it 10,000s, 10 millions of viruses? And what is then the diversity of the different cheats and the original virus in this load? But what role does this play with being infectious?
Yeah, this is such an important question, and there's a few different aspects of this that are important. So the first aspect is just the physics of it, right? Like how many particles are transmitted when an infection is started. And that is going to vary enormously depending on the transmission mode. So we know that for blood-borne viruses, for example, transmission from kind of direct blood to blood has a way wider bottleneck, way more particles transmitted than other routes. For things like coronavirus or flu, we think a lot of the transmission is aerosol-based, kind of droplets. And droplets has an interesting consequence, which is droplets closer to you might have a higher number of particles, but the ones further away are going to have a smaller number. And then the there's I get a distribution in terms of what's going to be transmitted. That's in terms of number of particles. The second thing is, what about in terms of the number of genomes that are actually initiating that infection? So the typical way that we estimate bottlenecks between for viruses is we look at the viral population and we infer the number of starting kind of genetic lineages that survived. Those estimates are typically really small, like 1 or 2 for most kind of respiratory viruses, and weirdly consistent among different transmission modes as well. What we think now, looking from the latest kind of animal studies where you can track kind of from very early after transmission, is that often what happens is a large number of particles initially infect, and then there's a very kind of severe bottlenecking event at some point, kind of in the first couple of days, which crushes the diversity, and then you only have 1 or 2 lineages kind of surviving from that. Now, all of that is thinking about it without cheats. There's 2 things, I guess, that are really important when cheats get involved. One is cheats. So as you say, if it's a bottleneck of 1 and it's just a cheat, the infection will fail. If it's a bottleneck of 2 and there's 1 cheat and 1 cooperator, I'm going to predict that the infection might fail. Because if these cheats interfere with cooperator replication, and we know that you have these severe bottleneck events, presumably this thing that has a massive fitness advantage but can't replicate is gonna be a problem, right? It's like trying to colonize an island when you're like 2 antelope and there's a lion on the island, right? It's not gonna happen. So I think there might be a causal effect there, whereby if there's high cheat abundances, actually the bottleneck, the kind of inferred bottleneck size might actually be smaller, because sampling a higher fraction of the population is more likely to get you a cheat, which should reduce the likelihood that that succeeds. So that's kind of the way I think about it. I don't know if any of those are answers to your question, but I think those there are kind of different components there, I think, where this matters.
Yeah, no, thank you. I have a follow-up question. Because you basically implied that the longer a host is infected, the higher becomes the frequency of the cheats. So that would mean that sick hosts, the longer they're infected, the less likely they are to infect other hosts. Can one test this empirically?
Yes. Yep, it does imply that, and yes. Now I guess there's 2 things. One, there's a confound with the immune system. So the viral cheats are spreading, but also as the infection goes on, the immune system is going to suppress the viral load. So if your infectivity goes down, it might not be that obvious what's causing it. The other thing here is, all else equal, a longer infection will lead to more cheats, particularly at the earlier time points. Right? This has to be true for the first couple of days, assuming that cheats are generated de novo. However, we know from the lab studies that what really happens with cheats is this kind of thing. You have these kind of these oscillations that I was showing. So it's possible that at a certain point for cheat cooperator interactions, you might start oscillating, and then there's a non-trivial relationship between age of infection and outcome.
Thank you.
So Asher, I have a couple of questions. I don't see any more in the chat for now, but I'm like Karsten. I feel like I don't know much about this, so if they come across as naive, they are. And also they're influenced because I give lectures at the 1000 level on viruses. So this is mechanistic. Do the different kinds of cheats of the cooperators interact differently with the host genome, thereby affecting sort of the cycling of the virus within the host? So I'm thinking lysogenic, transition-lytic. Would that be a potential factor that would influence the transmission and replication?
Yeah. Yes. So, okay. So for influenza, there's no evidence that any of these sequences are becoming lysogenic or getting into the genome or anything like that. We think that there's not a huge difference there. For other types of virus, yes, and there's potentially kind of an interesting thing. So, HIV. HIV, when it infects you, produces tons of what they call proviruses, right? Which are essentially deleted versions of the genome, the HIV genome, that hang out inside yours. So the vast majority of HIV kind of latent reservoir inside you is defective. It turns out that these defective HIV proviruses, some of them are cheats. There's some nice work from Robert Silicano's lab kind of earlier this year showing this. Now, there's like a behavioral ecology kind of lens on this, which might be what you're kind of thinking about, which is, if you're a viral cheat and you're free living, so you're just like in the cytoplasm, like the influenza ones, you kind of have this problem because if you're obligate, a cooperator needs to come along and like super infect, otherwise, or you have to be co-infecting with a cooperator, otherwise you die pretty quickly, right? You're going to get cleared. If you're inside the host genome, that's not necessarily true. If you're inside the host genome, maybe you can hang out, roll through cycles of replication, and then wait for a cooperator to come along at some other point, which would predict that these integrated viral cheats should essentially be much better at exploiting the cooperators. They should have like kind of longer timescales over which to adapt. And there's an interesting thing where in bacteria, a lot of bacterial immune systems against phages are basically cheat phages that integrate inside the genome and then like reshape the kind of lytic phage capsid to like spread in them and then re-lysogenize when they infect a new host. So you can think of that as a transmissible immune system, or you can think of that as a viral cheat that happens to be like in a defensive symbiosis with its bacterial host.
Wow. That's pretty cool. That made me think that does this then play into some of the different strategies or life histories of virus? Well, probably not the right word. The histories of viruses, like for example, shingles, things that hang out for a long, long time and then suddenly pop out again. Does this play a role in that kind of strategy they take?
Yeah, presumably. I mean, one thing you might predict is that if you're a cheat, there's a stronger selection pressure to integrate than if you're the cooperator. You know, whether that's under your control or not is not obvious, but if it is, you might predict there's that selection pressure. Therefore, maybe you're more likely to have a latent reservoir. In HIV, it turns out that sometimes when you get kind of reactivation and you get, in particular, and kind of retroviral resistant reactivation, sometimes that can come about through recombination events among proviruses. So there can be this kind of like longer term effect on the population that can have consequences for reactivation if you have this drive there. In terms of whether the like viral life history itself is shaped by for like the cooperator virus, I don't know, and I think it will depend. That kind of thing, I think, depends a lot on the between host cost of cheats. Like, you know, if you create these things and then they spread, but you've transmitted before they spread, maybe it doesn't have that much of an impact on your genome or your life history. Whereas if you create these things and then you have to contend with them, there you might want fancy life history strategies to avoid them. Like maybe, yeah, like all sorts of things you might imagine.
Yeah, interesting. I don't see any other questions, but I have another if it's okay. So one of the slides referred to Hamilton's role. I think it was a conceptual slide you had,
Yes.
and I'm curious, what role do you think that plays? You know, the kin selection plays, and also, and maybe I'm missing the target entirely. If you have these multipartite genes, if they're interacting differently with the host, does that change the relatedness essentially, which then affects kin selection? Does that make sense?
Yeah. Yeah, and so multipartite gets hard to think about. I think the way to think about multipartite is more like an egalitarian transition. So I think when they were all monopartite cooperators, it was meaningful to talk about relatedness between them. But by the time you encode gene 1 and I encode gene 2, I think we're more like mitochondria and nuclear genome. But for these other cheats that are being generated de novo, yeah, so I'll give you so one example where people have looked really explicitly about Hamilton's rule in viruses is this paper from Pilar Domingo Calap in Rafa San Juan at Valencia. What they did there is they had a mutant of VSV, Delta 51, which turns out to be a viral oncolytic therapy candidate. And the reason it's oncolytic is because it's a cheat, basically. So Delta 51 doesn't block interferon. What that means is if firstly, it means it can infect cancer cells, but not immune competent cells. But secondly, interferon blocking is a viral public good at the between host level. Because if we both infect cells and we both block interferon, we can continue infecting. But if I choose not to block interferon and you're still blocking it, you have a slower growth rate. You're keeping the interferon response contained. I'm growing a bit faster, and my cell is then alerting everybody else to the interferon so that we should then we both pay a cost. Now, it turns out the group cost of this is like enormous. So we're talking like log reductions in population growth, but nevertheless, there is a cheat advantage. So what they do is they vary the spatial structure of the population, and they use trypsin, which essentially detaches the cells from the surface to make like an R equals 0, like fully mixed case. And then they have the like well mixed case where they don't do that, and R is about a third or something. And then they actually calculate the C's and B's for Hamilton's rule, and they find a condition in which it's stable because R is above 0.25, I think is the critical value. And then the other one where it's unstable, the cheats invade and everyone does terribly. So that's a really nice example of them doing Hamilton's rule. I think one difference, which is what I was getting at earlier with this cheat advantage and cheat origins. A lot of the time in social evolution, we solve these social dilemmas by saying, oh, well, relatedness is high, therefore you keep interactions among kin, which is great, except in viruses, these mutations can happen so rapidly that even if your population is clonal now, I think there's a pretty good chance that it will not become clonal purely through mutation pretty quickly, and these B's and C's are then pretty extreme. So it's not obvious to me, particularly at this like within host level, if our kind of like equilibrium analysis approach using tools like Hamilton's rule can help us predict too much at the within host level, at least for things like flu.
Incredibly cool. It's Linda, Lonnie, and Carson. Do you have any other questions, the two of you, or comments at this point? We're just hanging out. In fact, I'll stop the Zoom.